This is the phase 1 clinical trial data. In phase 1 clinical trial, they recruited around 48 people. They used 4 arms, that means 4 protocols. In arm 1, they used 1 mg of DNA, and they used needle to inject it, and in arm 2 they used 1 mg, and in this case they used needle free system. And in third arm they used 2 mg, and in this case they used needle to inject it. And fourth arm they used 2 mg also, and in this they used needle free system. And you can see most of the adverse effects are found in first dose. Like in preclinical, here also they used 3 doses, 0, 28 days and 56 days. Most of the adverse effects are found in dose 1, and after third dose they found some mild effect, itching. So this shows that whatever the side effects are present in first dose, second dose and third dose are very much tolerable. Next, they checked the neutralizing antibody concentration. This is the neutralizing antibody concentration. You can see they have compared their neutralizing antibody concentration with naturally available antibody concentration after COVID infection. This protocol, which has 2 mg of DNA and also injected with a needle-free system, has maximum antibody concentration, which is comparable with natural system. And this is checked at 84 days. And also they checked IgG concentration. This is IgG. And you can see at day 84, the titer is maximum, and as compared to other arms, other protocols, the protocol which has 2 mg of DNA and also needle free has the maximum amount of IgG. So this shows this protocol is the best. Next comes, like in the preclinical data, they checked interferon gamma, but in this case they isolated PBMC, and to check the cell mediated immunity they performed a spot forming assay, and they found, you can see here, this is the arm, fourth arm, which shows very good response, cell mediated response. This also proves that the fourth arm, which is very much effective for producing cell mediated response and also antibody production. After the phase 1 clinical trial, they performed phase 2 clinical trial, which they recruited around 1000 people, and also from phase 2 clinical trial data they found this is very much effective against COVID-19. Although they have not published phase 2 clinical trial data, but they found that from phase 2 clinical trial they found that this is very much effective against COVID-19. So they performed phase 3 clinical trial, and recently they got emergency authorization use from government of India. This is from Ministry of Science and Technology, and they say that they recruited around 28,000 volunteers, and from this trial result they found that the efficiency is around 67%. Next, we'll discuss the advantage and disadvantage of this DNA vaccine. So first advantage of this vaccine is this is around 60%, 67% effective against COVID-19, and this 67% effectiveness comes against Delta variant, because the recruitment and the clinical trial when happened in India, that time it was the second wave, and we know that the second wave in India is because of the Delta variant. So this effectiveness is against the Delta variant. So that is a positive sign that this is also working against Delta variant, and this is very much inexpensive, because this is a plasmid DNA and it can be produced with less expensive way, and they are using needle free system that is also very much less expensive. And DNA is very much stable as compared to other viral vectors and also mRNA. So this can be stored at room temperature for around 3 months. And this has high production rate, because right now we need so much dose of this vaccine. So it helps because this is produced in bacterial cell, so this can be produced with less time, and this can help for the need of vaccine, urgent need of vaccine with large amount. And another important thing is this uses a needle free system. So who have the needle fear or needle phobia, this will be helpful for those kind of people. So this is also a good news. And what are the disadvantages? Because in this vaccine, it has to use 3 doses. So this takes longer time as compared to 2 doses, and also this will increase the transportation problem. So because this will need more amount of dose which needs to be transported. And till now, because we do not have all the clinical trial data, so this is also a negative sign. Overall, this is going to help the fight against COVID-19. So this is all about ZyCoV-D, the first DNA vaccine against COVID-19. Thank you for watching my video. See you next. Bye bye.